Okay, so this is learning assignment 18B, and I've got a Punnett square here for you. And what I was just saying is that in order for two parents who don't have a disorder to have a child who has a given disease or a disorder, and given the information here, we know that it's recessive. That means they have to both be carriers. And if you're a carrier, you are a heterozygote. Your genotype is that you have one allele that is dominant, that doesn't code for the recessive disorder, and one allele that is recessive that does record for it. And so the gametes for the parents are big T or little t. And so here we'll put one of them up on the top, big T, little t for the options of, you know, the egg, and big T and little t for the options of the alleles for the sperm. And remember, each gamete only gets one chromosome, so it's going to get one or the other. And what are the recombination options? Well, if both the egg and the sperm are big T, then the offspring will be big T, big T. If it's the half chance that the mom, it's the mom's dominant allele and the dad's recessive allele, then it's going to be big T, whoops, wrong spot, big T, little t. And if it's the mom's recessive allele and the dad's dominant, then it's big T, little t. And if it's the half chance of mom's recessive and the half chance of dad's recessive also, then it's little t, little t. And so inside are all the possible genotypes of the children. And you might say, well, why do I have big T, little t twice? And it's because these are the ways in which the alleles can combine based on the random fertilization. And that means that your genotype ratio, and I'm just showing you what this looks like, is 1 to 2 to 1. And remember, that's big T to big T, little t to little t, little t. And the phenotype ratio is, now look at it. How many of them are going to have Tay-Sachs? 1/4, right? So it's 3/4, these two up here, and this one down here, 3/4 that are at least have one dominant. And so the phenotype ratio is 3 to 1, where they have no disorder to having Tay-Sachs. So a quarter of them, or 25%, would have Tay-Sachs. So basically there's a 25% or 1/4 chance that any given child will have the disorder or disease. So that's how that one works out. Now, that's strictly Mendelian genetics, dominant and recessive. The chickens are not quite so clear. So if you look at this here, when chickens with splashed white feathers are crossed with black feathered birds, their offspring are all slate blue or blue Andalusian. Hey, there's something that is neither white nor black, nor white, some white and some black. There's something that is completely different altogether. Does anybody remember what that is? What we call that? Here, I'll pull open the PowerPoint here. I can get my mouse to cooperate.
Incomplete dominance.
That's right, incomplete dominance. Excellent. My mouse was slow enough, you got it. Okay. So, do you remember these slides here? Complete dominance is what we were talking about just now with Tay-Sachs, okay? And incomplete dominance is this, that a heterozygote phenotype is in between the two homozygous phenotypes. And codominance is different. It's when both phenotypes are expressed. Now, blue is neither black nor splashed white. It's something else entirely. So we said that that was incomplete dominance, okay? But can we explain then what must be happening, okay? So if the parents are white and black, then we know that just like the example that I had going here of the snapdragons, see how both snapdragon parents are true breeding? So that means our chickens have to be true breeding. I can't even talk. They're true breeding, not just true breeding, right? So the white chickens are going to be, should we use F for feathers?
Oh, so it can be true breeding and incomplete dominance, because I just crossed out true breeding.
Yeah, true breeding just refers to they have two alleles that are the same.
Yeah.
Okay. So true breeding just means that they're homozygous for what they have. Okay. And so if white chickens were crossed with white chickens, all you'd have is white chickens. If black feathered birds that were true breeding black feathered birds were crossed with themselves, all you would have would be black feathered birds, okay? So the white chickens, and I'm gonna come back here because this is gonna get fun where I want to insert, I'm gonna do a W here, and then I have to tell it. Will it let me do it here? No. I have to remember where in Word. Okay. To make it a superscript. Okay, so let's make this a little bit bigger. Okay, and the black chickens, and do you know why I'm using the capital letters? Because they are both going to get, it's incomplete dominance, so none of them is recessive, which means I don't want to use lowercase letters, and I'm showing them superscripted like this to demonstrate that. So if I put in, it's not going to let me copy the table. All right, I'll drag it. I'm just trying to make sure. No, it. All right, we'll stop trying so much to do. I'll draw a new table every time, I suppose. All right, so now if the white chickens make eggs or sperm, then we know it's going to be like this. And black chickens, it's going to be like this. So what happens when they mate? Well, I'm going to put one of each of these alleles in every box in between, because those are our only choices. A true breeding parent can only give one of the same kind of alleles. And so all of their offspring will be what? They'll be heterozygotes. They'll have one allele for white and one allele for black, but because it's incomplete dominance, this combination gives you blue feathers, okay? And what happens when the blue Andalusians are crossed among themselves? So that's the next piece here. So let me grab another table. And now we take these two, we take two blue feathered chickens and, oh, that is so weird. Why did it just wipe it out? I have no idea what just happened. So let's try this again. Oh, that is really frustrating. You see how it wants to get rid of it? Okay. I'm just messing with the table a little bit and trying to figure out why it doesn't like me right now. Oh, here we go. It's like I had two overlaid on top of each other. Like many, many copies. I apologize. Okay, so here's my original, and now I'm gonna change this here because we have the two chickens that are the blue feathered ones. So this is cross 1, and then this is cross 2 for my F2 generation. And so if you look at this, 25% of the time, I'm going to get chickens that just have white feathers, and 25% of the time over here, I'm going to get a white egg and a black sperm. And then another 25% of the time, I'm going to get a white sperm and a black egg. And then another 20% of the time, I'm going to get a black egg and a black sperm. So now what do I have genotypically here? Can you see that I have a genotype ratio that is 1 to 2 to 1 for, you have 25% of the time you get this. Oh, that didn't work out. Let me see if I can snatch that better. Okay, and then 50% of the time, you have this, and then 25% of the time, you have this. And it's getting, I'm not quite sure. Well, those dots are going to be a little bit higher because of the superscript, but that shows you the 1 to 2 to 1 ratio. And the interesting thing that's happening here is you also have the phenotype ratio is, well, what are these guys that are FW? They are white. What are the ones that are, oh, that's, it copied funny. Should be like this. The ones that are FB, those are blue. And the ones that are FB are black. So you actually also have a phenotype ratio that is 1 to 2 to 1 of white feathers to blue feathers to black feathers. And so that explains how you get the 1 to 2 to 1 ratio. It's in the F2 or the second cross from, you know, the homozygous true breeding parents if it's incomplete dominance. Ready for number 3? So this one says, in humans, the condition for normal vision dominates color blindness, and both alleles are linked to the X chromosome. So what does that tell you? It's an X-linked trait, and that therefore we're talking about something on the X or the Y chromosome, and it just gives it away here that is on the X chromosome, okay? So if a colorblind male marries a colorblind female, what do we say is happening there? So we know we've got XY for the male, and XX for the female. And if we want to use, remember it's if he's colorblind and normal dominates colorblind, then we know that it's recessive. So we're going to use a little b for colorblind, like that. And of course, that's going to be way too small for you to see. And if she's colorblind, then we also need her to look like that, okay? So in order for her to be colorblind, she has to have 2 recessive alleles, because if she gets 2 normal ones, then you know that she would not be colorblind, okay? So if they have a daughter, what's the chance that the daughter will have normal vision? Well, look at all the X's. They all code for what? They all code for colorblind. So will they have a daughter that isn't colorblind?
Mm-hmm.
Okay, so daughters are 100% colorblind, which is the same as 0% normal vision. You don't even need to do a Punnett score for that, because you can see that all X chromosomes are recessive for color blindness. Okay, and what about the sons? If you look at this one here, what is the only thing that the mom can give?
Little b.
The little b, okay? So the sons are all gonna be this, and they get a normal Y, but that doesn't determine color blindness, this does. So basically the sons are 100% colorblind and 0% normal vision also. I want you to look at this for a second and see if you can figure out what parent they might need to have so that girls would not be colorblind, okay? And I'm gonna go send my daughter's friend home. Take a look at that. So, did you figure it out?
No.
All right, so my question
The mom cannot wait.
is,
Hmm.
so for, what did I say? For daughters to have normal vision, which parent needs a normal X?
Wouldn't it have to be the mom?
For maybe I should qualify this, for them to for sure have normal vision, not maybe some of them. So we'll say to always have normal vision. So here, I'll show you.
I think it's got to be both.
Yeah, maybe.
Okay, so if this is, here, I'm just gonna copy paste. Okay, doesn't help if you copy the wrong thing and then paste the wrong thing. Okay, if this is mom, then what's she gonna give to everybody?
An X, a recessive gene.
A recessive gene, right? And then
Mhm.
remember, here's dad, and dad's gonna give this, so that means his sons, if mom has 2 recessive alleles, then the sons will always be colorblind, okay? But if dad,
Okay.
if dad is what?
He's still giving.
Okay, but what if dad is this?
Well, you said which parent?
Yeah, which parent, for the daughters to have normal vision, which parent needs a normal X?
Oh, I thought you said
Okay.
which one was to
Which
change.
parent would need a normal X in order for the daughters to have normal vision? So if you look at this, if dad is normal, then the daughters are always what?
Normal.
Then the daughters are always normal, okay? Looking at an
Okay.
X-linked recessive trait. Now I'm going to show you, and the reason I'm doing this is this will help you understand. Oh, oops, I just embedded it in the other table. That was dumb. Let's try this again. Okay, so now let's say that mom is normal, but a carrier, okay? And dad,
Normal. Okay.
see why she's a carrier? She has
Yeah.
a recessive allele, but in order
Mhm.
for an X-linked recessive to be shown in a woman, she has to have two recessive alleles, because otherwise this dominant one will block it out, okay? So the mom is not colorblind, but what happens to her kids? So here we go with daughters that
They're
are
fine.
fine.
Normal.
And now we have, but the other half of the time, she has daughters that are
Oh.
colorblind. And then what about the sons?
Fine and colorblind.
Fine and colorblind. So what happens here when you're thinking about patterns in families is if the dad has the recessive trait, then if the mom is a homozygous dominant, none of the kids will ever get it, okay? Because I could show you one more, I could show you one more box. I'll show you what I'm talking about. One more table, okay? One more scenario of this. So if dad is colorblind, so he has the recessive allele, so he's either gonna give his daughters a recessive X or his son's the Y. And if mom is normal, like, and not a carrier, then what will always happen? The mom will always give a normal X, right? All the time. And so all of the kids will be fine.
Mm-hmm.
But if the dad is normal and the mom has it, then the daughters are fine, but the sons will all have it. So
Weird.
when it's X-linked recessive and the mom has it, the sons always get it because it's on the X. So mom will pass it to the sons. But the daughters get half of their X's from their dad. And so if he has a normal X, then they always get a normal X. And they don't have it, but they become carriers, okay? And a carrier means that you're a heterozygote, but you don't have the disorder or the disease or the trait that's less desirable, so to speak. Maybe it's fun to not see red and green.
Is that like rule across the board, or is it just a good idea to always do the cross?
If you know what's going on, you don't have to do the cross, but I'm just showing you how to understand X-linked traits better beyond just what I asked in this question. So this isn't, none of these are what was given, but this helps you to see how it all works. So if the dad is colorblind, and the mom is not colorblind but she's a carrier, then their girls are normal but half carriers. Sorry, I need to go this way. Their girls are normal but half carriers, and their boys, I just said that wrong.
Mm-hmm.
The girls are half carriers, half affected, and the boys are half normal, half affected. Because the dad's always going to give the affective recessive, and the mom's going to give it half of the time. So half of the time, you'll have that affected dad's X combined with the mom's affected recessive X, okay? And so it's just interesting to see that when the mom is affected and the dad is normal, then you see affected sons, but you don't see affected daughters. Okay. And that up here, basically, it's when, you know, mom and dad both have a recessive, then everybody gets it, right? And that makes sense anyway. Any recessive thing, if both parents are homozygous recessive, then all their kids would be true breeding also. Okay. All right. Blood type A, B, and O. We talked about this last week. So let's see. A and B are dominant over type O. And a person, this one says, a person with alleles for blood types A and O marries somebody for B and O. And what does that look like? Now remember, here, we're gonna do IA, I like this, with somebody who is, and if you remember last week, what I was showing you here was that we show the the blood type with a superscript also, okay? And for the O, they just don't make the protein, and so it's the little I showing the absence of the protein. So somebody who is A but only has one allele for A marries somebody who is B but only one allele for B, okay? And what are the types of blood their offspring could have? So you can try, this is where you can do FOIL or you can do a Punnett square, okay? So if we did it FOIL, then you have to do first. So IA, come on, and IB are the first alleles in each set. And inner would be this first one and the second one, sorry, the second one on the first parent and the first one on the second. And we always put the one that's dominant first. That didn't go very well. Try that again. It doesn't want to do it. My computer's decided that that whole spot needs to be a superscript space. And so I'm having to convince it that it's not. So this would be the second version. And then outer would be
IA superscript with a lowercase I.
And then this.
2 lowercase i's.
Okay, and then the last one would be the last, like that. And can I get the same thing in a Punnett square?
Mm-hmm.
Yep. So then the way I do that is I have to say, all right, this is mom. Well, it just says a person, it doesn't really matter, with blood type. Here's one person. It matters whether or not they are male and female in order to have, you know, to successfully have eggs and sperm. Because last I checked, an egg could still not fertilize an egg. So we got to have eggs and sperm here. And so what are their combinations? And you just put them in the middle of the Punnett square. So you got the gametes on the outside, and you have the zygotes in the middle. And I don't know if that helps you to track a Punnett square, to think about that. Gametes on the outside, combined fertilized zygotes on the inside. And so then here are my 4 different combinations. And what is the probability of each of those blood types? Well, they all show up once. And so if they show up 1/4, then it's a 25% or 1/4 chance for each blood type, okay? So thinking about that and what you see here, in which family might you question the paternity of a child? And you don't question the mother as long as we're assuming there was no in vitro fertilization. We're assuming that the mother is the biological parent for sure. So why would, which one of these fathers, the one with the A blood type or one with the AB blood type, be in question, okay? So can a mother and a father that are A and O have a child that is either A or O? Remember, what if the father is actually this? And if the mother is O, then we know that she is this. So you can have IA combined with I to give type A, and you can have I combined with I to give type O blood, okay? Can we get in the second instance, sorry, I'm trying to make that paste thing disappear. In the second instance, can we get a type O child and a type A child from an A or a B parent? Now, if the parent was IB, that wouldn't work. But what if the parent is this, okay, and the other parent is this here? So if and if you look over it here, can we get a type A? Yep. Can we get a type O? Yep. So family 2 is okay, but now look at family 3. If this person, if the dad is AB, he gives her A or a B. He can't give what? He can't give an allele without a protein that codes for an antibody. He doesn't give O. So if they have an O child, it's either a new mutation or not his kid. Because they can have kids that are A, or they can have kids that are AB, but that's the only option here. Oh, actually, if this is what she is, then they could also be, then they could have a kid that was just type B with an O, but they can have A or they can have AB or they can have B, but they can't have O. So this is not possible.
Red flag.
All right, so let's see. Number 6, neither straight hair nor curly hair is dominant. So that's giving you a clue right there, non-Mendelian genetics. Heterozygotes have intermediate phenotypes. Mary has wavy hair and reproduces with a man who has wavy hair. They have a child with straight hair. And what are their genotypes and what are the genotype and phenotype ratios? Okay, so when we're looking at this here, we know that we're going to have to use that superscripting again because there's no dominance here. And the kid doesn't have like chunked straight and chunked wavy hair, so they're not both being expressed. So this is another example of incomplete dominance, okay? So that means if we're looking at hair, right, then let's see. Mary has wavy hair and the dude doesn't get a name. So let's see.
I named him Bill.
If they're incomplete dominance, then one has to code for curly and one has to code for straight. And then let me superscript this and blow it up for you so that it's bigger. Okay, so both the parents are this, okay? because they have one allele that codes for curly and one that codes for straight, that together give an intermediate or wavy hair. And actually my family is a good example of another way this plays out. My husband has straight hair, I have wavy hair. 2 of our children have straight hair and 2 of them have wavy hair. So my husband only gives the straight hair gene or allele, you know, that's the only one that he has. But half the time so this is me, and half the time I have given a curly one and half the time I've given a straight one. So half of our kids are HSHS, straight hair, and half of them are HCHS, and so they have wavy hair. But if you look at this combination here, so this is mom and dad, and if the kid has straight hair, what does the kid have to be? So these are the parents, and we know that the kid has to be this. And it says, what's the ratios for all potential offspring? What does that tell you that you have to do if you can't remember, or if you can't figure it out without a Punnett square? Then you have to draw one. Okay, so here's mom. And here's dad. And how do they combine? Like that. And so what's my ratio here? Well, I have a 1, and here I could highlight it. I wasn't thinking about that. I'll show you with highlights. No, not like that, please. All right, see how I've highlighted them for you? So 25%, and they have a 1 to 2 to 1 ratio. That's the true for genotype and for phenotype. So their genotype is 1 to 2 to 1 as seen here, and their phenotype is 1 to 2 to 1, 1/4 of the time curly, 2/4 of the time wavy, and 1/4 of the time straight. Does that make sense? You're all good? You're all good now? So I'll write this out. Both genotype and phenotype ratios are okay. And the phenotype is 1/4 curly to 1, well, I'll write it like this, 2/4 or 1/2 wavy and 1/4 straight. Okay, and the 1 to 2 to 1 genotype is as you see that I've highlighted in there. Okay, so I'm going to close this, and then I'm going to hope that my daughter doesn't start playing with the noisy helicopter, but she is. And what I want to do is highlight, oh, this is part 2. I want part 1, and I haven't managed to grab it. Yes. I just wanted to, I highlighted a couple of things here. And I just want to take about 10 or 15 minutes to answer any questions you might have right now. So does anybody have anything in particular?
Just briefly, on the very last problem that we did, it said intermediate, what did it say, intermediate heterozygous, and I just, that intermediate part.
Oh, intermediate means in between, neither one nor the other. So it says neither straight hair nor curly hair is dominant. So it's giving you a clue, and that the heterozygote doesn't have curly hair strictly or straight hair strictly. So I have wavy hair. People might say it's curly, but it's not spiral curly. It's in between. So I have a straight hair allele and I have a curly hair allele that together, you know, I have some proteins that code for specific actually disulfide bonding in my hair that makes it slightly curly, which we call wavy. And so intermediate means neither one nor the other. So neither curly nor straight, it's in between.
That makes sense.
And another thing that we call that in genetics is incomplete dominance.
Okay, perfect. I'll make a note of that. Thank you.
Okay, you're welcome. It's how to give you a hint about what it is without telling you what it is, so that then you have to identify it. That's why it's stated that way. Okay, so there are two sections here that I mentioned last week with genetics. And I believe I talked about, hey, focus on looking at, you know, what did Darwin do? What are the main things that he figured out? And then coming through and looking at the original evidence for evolution. So I wanted to just highlight a few of these slides. And on Thursday, half the time we'll do the second set of evolution slides, and then half the time I'll do some like Q&A review for anything from earlier chapters, okay? So this slide I wanted to talk about, it probably makes sense to you that if there are beetles that are, you know, have variable carapace color based on their proteins that code for the pigmentation, that if they're living on a habitat that has a darker background, then the ones that are the darkest would be the most easily camouflaged. And so which ones are going to be the most likely to get eaten during their lifetimes? The ones with the lighter pigmentation. And so you can see the bird picking them off, and then what happens is the survivors reproduce, and what do they give to their offspring? Some of them are going to have lighter alleles, but the majority of them are going to have the darker ones. And so what happens in the population is that the offspring are darker overall than the original parent generation. And so as the bird eats beetles, and we go through successive you know, matings and new beetles being hatched, each generation is a little bit darker as far as the overall alleles that exist. The total number of alleles that are dark goes up compared to the total number of alleles that are light going down. And we call this natural selection, where something in the environment selects for a particular allele over a different version of an allele. And there have to be alternate versions in order for selection to occur. The bird wouldn't have a choice if everything was dark or everything was light, right? It would just catch what it could catch, and it would all be the same. And so, you know, Darwin noticed this, and it's one of the biggest things that was the basis for his evidence of evolution by natural selection. And people who were already noticing differences in embryos, differences in fossil structures, and starting to, you know, be open to the idea that organisms could change over time, that there was evidence in the fossils, that there were evidences in, you know, living organisms, that things in successive generations weren't exactly the same, but nobody had come up with an explanation for how that could be possible. And so Darwin basically came up with the term that we identify as natural selection today. And before him, it was totally bogus. It was, you know, you might call it malarkey, but somebody named Jean Lamarck proposed that organisms could change because they wanted to. So that, you know, we get what we want out of necessity because we need it, that somehow need creates the change in genetics, or that by achieving something by exerting our muscles or by exerting our brain, we could somehow pass that on to our offspring. Wouldn't it be nice if my parents learned how to earn money and they could just genetically pass on that knowledge for how to earn money to me? It would be nice, but it's not actually possible. There are things that we teach our children that we have learned, but we teach them through instruction, not through our genes. And so his premise that a giraffe, you know, the reason that giraffes are taller now than they were thousands of years ago is because the trees got taller and there was competition and it was harder to get the leaves. And so the giraffe wanted to be taller, so it stretched its neck. And by stretching, it got a taller neck, and therefore it had babies with taller necks. It was proven to be false, but it opened the door for people to be able to consider change over time. And that was the gift that Lamarck gave us. So we can disprove his theory of evolution by experiment. We have many times over. We know that there's no such thing as acquired characteristics. Anything that you acquire in your lifetime through effort, you don't pass it on genetically. The only way you can pass it on is if you teach your offspring directly. And so there's no innate tendency toward increasing complexity. There's no innate tendency in genetics towards perfection. There's modifications in genes over time based on random mutation and what the environment then selects as advantageous or disadvantageous. So that's just kind of sets things up here for you. So watching the Bozeman science video on the history of evolution would be really eye-opening. But I wanted to just show you that when Darwin went on his worldwide cruise, he spent a lot of time studying the Galapagos, and that's one of the things he's most famous for. He definitely noticed the island organisms were different than mainland organisms, but then he gets out to the Galapagos and he sees that there are all kinds of finches, these birds that all have these, pointy beaks, but some of the birds are bigger and some of them are smaller, and some of them have pointier, skinnier beaks, and some of them have deeper, thicker, stronger beaks. And he paid attention to what they were eating. And some of them ate insects and little seeds. Some of them ate flowers. Some of them ate big seeds and nuts that they had to crack open. And he noticed that the habitats on those islands was not the same, and that the trees and the vegetation produced different kinds of forage for those birds. And basically, you could think of if a set of birds got blown from the mainland and hit this first island, and they survived and they reproduced, and there was a certain kind of food for them, that the ones who were best able to eat that food would grow the biggest and have more finch kids, right? And so that over time, the vegetation would select for the beaks that were best for eating that vegetation. Well, what happens if some birds from here get blown over to this deserty island over here and the vegetation is different? Now the beaks that they have, if the beaks here were for like flowers and seeds, but over here there are big dry nuts, now who gets to live out of like a group of 50 birds that gets blown over here? The ones that have the beaks that are bigger and deeper have a better chance of getting food and living long enough to have offspring. And so it's their randomly bigger beak genes that are selected for. And so what happens to the population that grows over here? Their beaks over time are different than the beaks of the birds that live over here. And it's not because the birds that flew over here were like, yeah, we need a beak that's bigger, so we'll just go ahead and grow one. You don't get that chance. It's that the ones that could eat the food survived, and then they got to have offspring. And so that's how all these different islands got colonized, and why we see all these different kinds of beaks. It's based on the vegetation, the habitat that is provided. And there's this couple that went back and studied, Rosemary and Peter Grant. They basically spent like half their lives on the Galapagos studying finches. And they actually studied the finches during some drought times. And essentially they noticed that when there was a drought and the amount of water went down, what happened to the vegetation? the seeds became less available, the population size plummeted, and the birds that were better at getting through those thick, dry seeds were selected for. And so within a couple of years, they actually saw the population changing. And they saw it every time there was a drought. There were like 3 droughts while they were studying the birds. So really cool confirmation that Darwin wasn't a complete wacko, and that what he saw with the finches was accurate. Now, in Darwin noticing things in his journeys, how long was I talking to myself? 3 or 4 minutes?
Yeah, about 3 or 4 minutes.
All right. How much of the bird stuff did you get?
Started talking about the beaks, and then there was a drought, and then the bugs were gone.
Okay. So
And so their beaks.
I
got
was talking
thicker.
about, was I here?
Yeah.
Yeah. So had I finished talking about the birds getting blown over here?
Yes, ma'am.
Okay. All right. So basically the summary was that if the different vegetation types vary, then the birds that get blown over to that island that have the beaks that are best able to take advantage of the food available, those are the birds that survive, and those are the birds that reproduce and have offspring. And that's where survival of the fittest comes from. And so then the bird populations change over time based on the environmental pressure that selects for beaks of a given length and depth and size. And so Darwin used that to help him come up with these 5 key points. He'd already noted that, hey, most organisms reproduce differently than humans, so they have way more offspring than actually survive. And those organisms, those offspring that they have, are not all clones of each other. They're not all the same, so they vary. And this is really critical, that some of that variation is not just based on the environment selecting phenotype, but some of it is based on genetics, and some of it is heritable. And remember, Darwin didn't know about chromosomes and mitosis and meiosis either, any more than Gregor Mendel did. And so he said, okay, if some of the variation is heritable, then that's going to influence how many offspring a given organism has in a population. And so that's going to change how the genes of the different parents are reflected in the population that is made from their offspring. So the number of kids you have and the particular traits that you have are going to, be present in the next population. And he basically figured that based on these key points, that speciation must necessarily follow. And he'd actually come up with an explanation for how evolution occurs, but it's what we call microevolution, changes in a population's genetics over time. And it doesn't necessarily give rise to a new species. It can just change the population over time. So the finches, definitely we see different species, because if you bring those organisms back together, they can't successfully reproduce and create viable offspring. And viable means offspring that can survive and grow up themselves to mate and have offspring. And he proposed natural selection as a way that evolution occurs in change in populations over time, but he actually didn't have an explanation for speciation. And he made assumptions about what our ancestors would have been like based on what he was able to observe now. And we can't go back in time and test, what was around 10,000 years ago, but we can go look for fossils. And so that gets me to, some of the original evidence that helped people believe that there was change in organisms through time was looking at embryos, looking at vestigial structures, things that we have that seem useless. So why are they there? Were they around for some purpose in the past? Homologous structures, which show that show common ancestry, and then looking at the fossil record and biogeography, and then DNA came later. So embryology is just, in a nutshell, it's looking at early embryos and seeing that they have structures that are similar, and that in some types of organisms actually disappear later, but just seeing, hey, when we look at development, there's a lot of steps and processes that are similar. So it's a hint that development, if development occurs similarly, then we must be related somehow. And in fact, we now know that all organisms use, you know, when we're talking about living cells, we're talking about DNA as the genetic code. So it makes sense that DNA codes for proteins, that we would have some similar structures. Vestigial structures, as I mentioned, refers to structures that are useless. So like, what good is it for you to get scared half to death and for your hair to stand on end? It's cool for the people who freak you out, but all it does is give you goosebumps. But in other organisms, if you've ever seen something make its hair stand on end, it's still effective. But for humans, it's pretty much a useless trait, and so we call it vestigial. We still have it, but it's useless. Another example is when we look at a whale embryo, we see hind limbs, and then when you actually look at, you know, whale skeletons, you see a little bit of a limb structure that's left behind. Well, why did whales ever have hind limbs? And so that embryology is indicative of common ancestry. And then, and I'm going pretty fast through this because you have like 3 or 4 videos that you can watch that go through this for you, right? And I just, oh good, I still have the video panel hidden so that my daughter's not crawling around on video right now, just for your entertainment right now. So when we look at homologous structures, well, where did they come from? And when we compare the 4 limbs of vertebrates, the color coding shows that, hey, this bone and this bone and this bone and this bone are all the same. They serve the same purpose, but their structure has been modified slightly. But if all those bones are the same, as in when we count the bones, we see that all the same number of bones is there. They are in the same spot. They have the same, they have a very similar function. Then it suggests that there must have once been a vertebrate ancestor that evolved all of these bones, and since then, all of these different groups of organisms have radiated out from that particular ancestor. That doesn't mean we have fossil proof of the ancestor. It means that the structures themselves suggest relationships. And the converse of that is analogous structures, and there's a video to watch about that. So it doesn't mean that just because something has a limb that it's, you know, related to humans. So kind of fun to look at. So one of the slides I have in here is about adaptations. So if you have a mutation, like you just by yourself, you don't pass that on. What do you have to have a mutation in order to pass it on? It has to be a mutation in your gametes, and it has to be something that makes a change in the proteins, right? And if there is a change in the protein structure of the offspring, it can do nothing, can make no difference. It can be a harmful change, or it can be a beneficial mutation. And if it's a beneficial mutation that helps the offspring survive, then we call that an adaptation. Oh, my babe's trying to say adaptation. Okay. So the other ones in here are the fossil record and biogeography. And so I want to pull up this slide on the fossil record. And I want to show you that the trend when we look at fossils preserved in sediments over time is to go from simple organisms to increasing complexity through time, so that the deeper layers hold the oldest fossils, And they are much more simpler, and often, and when you go very deep, it's things that lived in calm inland seas that don't exist anymore, going all the way back. And then we see, changes in organisms that became increasingly complex. We see fossils of organisms that were able to live on land. We see fossils of plants, and when we go back and we age rock to several 100 million years ago, the oldest rocks don't have plant fossils, but the ones that are 300 million years old do have plant fossils. And for anybody who's listening now or listening later, this doesn't refute the possibility of an intelligent creator who set up the earth and allowed natural processes to take their course, just in case you were considering that. And so we have fossil evidence that the dinosaurs existed, but they don't now. We also have some evidence of a meteor strike that would have changed the atmosphere and made it so cloudy that things didn't grow. And what happens to all the plant-eating dinosaurs if there are no plants to eat because there's no sun to grow them? They die, right? And then what happens to all the meat-eating dinosaurs if there's no plant-eating dinosaurs to eat because there are no plants because there's no sun?
They die too.
They die too. And so a meteor impact that created a dust cloud that lasted decades would have killed out the dinosaurs. And volcanic eruptions could have played a role in that as well. We just celebrated 40 years of Mount St. Helens, so there was a lot of stuff in the newspaper, but we know that there were ash clouds that went to the north and to the east that changed things significantly. And actually back in the early 1800s, there were several volcanic eruptions that weren't in the United States, but nonetheless changed the atmosphere enough that it made farming in the Northeast United States really difficult for a decade or more, and actually is part of what motivated the shift for people to move, across the United States toward, what was the West then, which was, beyond the Mississippi, right? So the last one is biogeography. And if you take a look at Africa and South America, they actually appear to nest together really well. And if we date the rocks and we look at some of the mountain ranges and structures in some of the continents, the ages of the rocks are the same. And the fossils that are in those rocks are the same. And the patterns of the mountains can be the same. And what does that tell you about the continents? A long, long time ago, they used to be what? Stuck together. And over time, as the continental plates that hold the different continents have shifted and moved through, plate tectonics and continental drift, we have had the continents move and separate to where they are today. And the reason there are organisms that are on Australia and New Zealand that don't live anywhere else is Australia broke off first. And so it's been separated longer than any of the rest of the continents. And so Australia has had more time for, isolated evolution, by a natural selection, working on new mutations to occur. And the last thing, and I know I'm kind of rushing because I see what time it is with my little, 4-minute talking to myself. The last thing I wanted to talk about is what is not considered original evidence, but is the new and best evidence for evolution. And that is in DNA. So we just spent a couple of chapters looking at DNA and how the proteins that build an organism are coded for in your genes, in your genetic material. And so the DNA is used to translate nucleotide sequences into amino acids, and it's basically the same in all organisms. What makes one organism different than any other? It's the sequence of those T's and C's and A's and G's that code for the different proteins. But those proteins are all built from the same 20 amino acids. So the fact that we share DNA and that we share amino acids is a powerful argument in favor of having common ancestors and having, you know, common descent. Okay. And cytochrome C is another bit of biochemical or biomolecular evidence. It's a protein that is used in the electron transport chain, and every eukaryote, every multicellular organism uses the electron transport chain and has cytochrome C. And the cytochrome C can be compared. It's not the same across all types of organisms, but it changes a bit. And we can see how close the cytochrome C is from one species to another. And it helps us to make genetic comparisons with the DNA and with cytochrome C. And if you're watching my face at all and you wondered why I made a funny face, I didn't shriek, but my 1-year-old just bit me. So that's it for that particular PowerPoint. It's just considering the original evidence. So the learning assignment is just walking you through a few of those and having you consider how they are meaningful and what it actually means to be, you know, original evidence. And some of it isn't like we have hard and fast common ancestors. We have fossils that show relatedness, but we don't have an original organism that was the first, you know, the first or the earliest ancestor. So I'm not asking you to come up with anything like that. I'm just asking you to understand how all of these things show that all the organisms that live on the earth must be connected somehow if we all share DNA sequencing and cytochrome C. And what I'm going to talk to you about on Thursday is this evolution part 2, where we'll talk about the 5 mechanisms of evolution. So make sure you finished reading chapter 11 by Thursday, and it would be good for you to have both learning assignments done so that we could talk about them. And I'll make sure, and I'm going to pull up number 20, if it'll pop open right now. I'll make sure that part of what we do is talk through number 20 so that you know that you have it right. And it's due on Thursday night at midnight. So hopefully you'll have it done already by the time we talk at 3, but if you don't, you'll get some bonus help. Any questions now